Hello, my name is Rigdon Waite. I'm presenting a study titled Defective HIV-1 Proviruses Produce Viral Proteins. So this study was done in conjunction with many institutions and groups, namely the Laboratory of Immunoregulation, the National Institute of Allergy and Infectious Disease, National Institutes of Health, and many others. The study was submitted in October of 2019 and then contributed by Dr. Anthony S. Fauci in December of 2019. Before we dive into the meat of this study, we first need to identify and define a few key words and phrases that will help you better follow along and understand this study. To start off, we have the word that may be familiar to you, HIV. HIV stands for Human Immunodeficiency Virus. This is a virus that once infiltrates the body, with time passing, causes a disease called AIDS. It attacks the immune system by killing specific cells in the body that are tasked with ingesting and detecting foreign pathogens, rendering the victim vulnerable to outside compromise. HIV as a virus actually inserts its own genetic information into the genetic information held within your own immune cells. This means that it can hide exceptionally well. AIDS stands for Acquired Immune Deficiency Syndrome. It's important to note that HIV is a virus, not a disease. AIDS is a disease caused by the human immunodeficiency virus. AIDS is the disease that is caused by the slow leaching of your immune system by HIV. When delving into this study, you'll see many references toward a word called CART. That is C-A-R-T, and it stands for Combined Antiretroviral Therapy. There are specific drugs that can target HIV through various mechanisms. HIV, however, mutates quite often, rendering these drugs sometimes ineffective. As we combine different drugs together, the chances that an HIV virus mutating and becoming immune to this combination drastically decreases. We call the therapy of administering all of these drugs together combined antiretroviral therapy, or CART. The next on the list is PBMCs. This stands for peripheral blood mononuclear cells. This is just another label for specific immune cells. These are specialized cells in your body that detect and protect you from foreign pathogens. Immune cells can also detect antigens of the HIV virus. This means that any small piece of the virus can be detected and cause an immune reaction, rushing these specialized cells into the area the HIV proteins or antigens were found. The next on the list is HIV RNA. That stands for ribonucleic acid. It's a chemical instruction template that deals with how to build an HIV virus. This then gets read by various machines in a cell and then produces an HIV virus itself. Next, we have GAG proteins. GAG protein stands for group-specific antigen proteins. These various printouts of the HIV RNA help go and create a physical virus. However, GAG proteins alone cannot induce an infection. Right next, we have NEF proteins. These are another HIV RNA printout that are important for the creation of an HIV virus. Again, NEF proteins, just like GAG proteins alone, cannot induce an infection. They must be properly printed and combined with all the other proper printouts to complete an HIV virus that then can go out and infect other immune cells. The function of NEF proteins aren't entirely known. Next, we have CD4 receptors. CD4 receptors are specific receptors or sites that HIV uses to enter the cell and trick special immune cells called macrophages, which in essence eat and dissolve and show other immune cells foreign pathogens and foreign pathogen pieces. Lastly, a provirus is genetic information that a virus like HIV puts into the genome, or genetic information held within the immune cells of your own body. Awesome. Okay, amazing. So now that we've expanded your vocabulary, you're more likely to follow along with this study. Defective HIV-1 proviruses produce viral proteins. So as we proceed, there are some questions that further arise as we go into the study. Those questions are: what inspired this study to be done? What is the study hoping to discern? What does the study have that others don't? Who is the study in service of? What can be done with the results of the study? Well, in order to answer these questions, I took upon me some logical liberty due to the lack of explicit information given towards the questions by those who actually did the study. As we proceed, these questions' answers will become more obvious. So what was the study even about? Well, the scientists went forth to look into defective proviruses. These are proviruses that had been introduced into the immune cell by HIV, but despite years of CART are still sitting in the genome and being printed. They're defective because they have some mutation or deletion of genetic information that makes them, well, defective. Scientists were curious, however, despite patients being treated with CART and having undetectable amounts of HIV in the blood, they still experienced ongoing immune activation, causing inflammation, causal mortality, liver disease, renal disease, and cancer. So this study seeks to provide evidence indirectly that these HIV viruses are actually able to get involved with protein production by the evidence of induced long-term persistence of antibodies for HIV proteins in those on CART, and a persistent immune response towards the cells that are infected with these proviruses. So right off the bat, we can go ahead and look at the first 4 questions presented earlier. We can answer the first one by understanding what this study is about, right? So the defective proviruses and persistent immune response of the patients and the link to the persistent immune response despite undetectable levels of HIV have most likely inspired this study to happen. For question 2, well, the study aims to indirectly provide evidence that HIV proviruses are actually capable of printing out HIV proteins that can be recognized by other immune cells. And number 3, this study would most likely have not happened if others had already indirectly proved that these proviruses actually print out HIV proteins. So there we can already deduce it has what others don't. For number 4, this study is in service of those who are currently being treated through CART for HIV and are still experiencing active immune responses. These people infected with this virus go through so much. Anything that can be done to further our understanding of this virus is a miracle. Well, moving back to the study, how did they actually indirectly prove that defective HIV proviruses aren't, well, actually that defective? Well, they studied the natural ability of these HIV proviruses to create viral proteins in 2 ways. The first is using a standard line of special immune cells infected with HIV, called T cells, that HIV actually infect. These are termed H9MN cells. The second way was actually looking at clones of T cells that are infected with HIV from an actual patient being treated with CART. These 2 methods were used to analyze the expression of the proviruses, or the 2 methods that were used to analyze the expression of the proviruses were Western blots, used to identify and separate proteins, and confocal immunofluorescence microscopy, which is used to actually image the expression of viral proteins on photo through fluorescence imaging. So here we're going to obtain both qualitative and quantitative evidence. So now let's dive into the results for the first part. We can see here in the red box that the team was able to distinguish and divide the cells from the standardized H9MN line into 3 groups based on their expression of a specific envelope gene, protein, based on a high production, intermediate, and a low, compared to the defective and full intact viruses as shown in the second square. So this provides evidence that the cells producing the highest amounts of proteins are in fact the cells with complete proviruses. We're also able to see that the defective proviruses are also leading out with the production of this protein. Awesome. So here on this slide, the scientists broke up the groups of cells into 3 groups again. The first containing full-length, intact proviruses, with the second containing full-length defective viruses that have been frame shifted by 1 base, and then a short defective provirus that has a lethal deletion. So here, B and C in this image give evidence through Western blot that these claims in fact stand true. And then image D provides qualitative data for the purpose of actually seeing the expression of the proteins the full-length defective virus can show. This is the first part of evidence that these defective proviruses in fact do produce viral proteins. So moving on to this third part in this experiment, in this study, to provide these indirect evidences, they went out to gather actual clinical patient T cells infected with HIV and tried to find the provirus within those cells. So in order to gather these T cells and effective proviruses from a patient infected with HIV, blood was needed to be obtained, after which T cells were retrieved and cloned until 2 T cells had the same proviruses. After 2 months, they were able to isolate 2 clones that had the same defective provirus called P365. After further analysis, this ultimately was the predominant virus that existed within the patient. Through Western blot, it was easy to see that this was in fact a provirus with internal deletions. Lastly, through Western blot and confocal immunofluorescence microscopy, we can see that the GAG and F proteins are in fact showing up and being produced despite the provirus being defective. So there, through a standardized cell line and a clinical patient's cells, the study has provided indirect evidence that defective proviruses are in fact responsible for the novel HIV viral proteins being made. This expression of the viral proteins is what is causing the persistence of antibody response to HIV proteins and immune activation in patients who have been treated with CART to undetectable levels of HIV. So lastly, we have question 5. The last question finally appears. What can be done with this study? Well, hopefully this study can further develop into a treatment or possibly insight to a treatment that can further reduce the effects of HIV proviruses on those being treated with CART. This could serve well for those who are currently infected with HIV and provide an ease of some suffering for the victims of this virus and their families. So thank you so much for listening. I enjoyed putting this together. Have a good night.